Hello everyone, thanks for coming to listen to my presentation. I'm Guan from Shanghai Ten's People's Hospital of Tongji University. The topic of my paper is Radiotherapy Synergizes with PD-L1 Blockade to limit post-ablation cancer recurrence and metastasis. The outline of my talk as follows. Well, let's move on to the background. With the rapid development of ablation technology, patients with solid tumor benefit from ablation treatment for its safety, minimal invasiveness, and effectiveness. However, similar to surgical resection, the 5-year recurrence rate of ablation therapy could be as high as 70%, remaining a big challenge for tumor therapy. Growing evidence have shown that the immune TME plays a key role in tumor recurrence after ablation. However, the specific immune mechanism of tumor recurrence after ablation has not been clarified. Revealing the underlying mechanism is crucial for further intervention. The present study aims to elucidate the immune responses in residual tumors after incomplete microwave ablation therapy and explore effective treatment strategies against post-ablative tumor recurrence. To clarify the potential molecular mechanism of the chiral link between incomplete microwave ablation and local tumor outgrowth, we perform transcriptomic analysis using isolated recurrence tumors and untreated tumors. Gene ontology analysis reveals that the differentially expressed genes were mainly enriched in the immune-related biological process pathways. Further polychrome immunofluorescence and flow cytometry results displayed that incomplete microwave ablation induced occurred TME featured by hypoxia and MDSC infiltration, thus mediating the rapid recurrence of tumors after ablation. To address this issue, we developed a nanoparticle-assisted radioimmunotherapy strategy against post-ablation tumor recurrence. This nanomedicine is constructed by hollow manganese oxide nanoparticles loaded with PI3K gamma inhibitor IPI549. On the one hand, manganese oxide nanocarriers can in situ catalyze the decomposition of hydrogen peroxide into oxygen within the tumor site for enhanced radiotherapy. On the other hand, TME-responsive release IPI549 can effectively block the MDSC's trafficking and promote the transformation of M2 macrophage into M1 macrophage. The two complement each other to reprogram the post-ablation immunosuppressive TME into an immunogenic phenotype, thus exerting the maximum anti-tumor effect. TEM images clearly displayed the spherical morphology and hollow structure of HMP nanoparticles, which facilitate the loading of small molecular drugs. Notably, this nanomedicine possesses TME-responsive degradation profile and drug release kinetics until in vitro simulating TME. The cytotoxicity and hemolysis rate of the nanoparticles were negligible up to a concentration of 200 ppm. After intravenous injection, there was no significant difference in blood biochemistry indexes and major organ HE staining in mice compared with the normal group, indicating that this nanomedicine exhibits excellent biosafety both in vitro and in vivo. Since oxygen is a key factor affecting radiotherapy efficacy, we initially tested the catalytic oxygen production capacity of the designed nanoparticles. Rapid oxygen bubbles was observed using ultrasound in the presence of nanoparticles, and the oxygen level was proportional to the manganese concentration. Further double-strand breaks C and colony formation test results show that this nanoparticle is expected to be an effective radiosensitization agent under hypoxic TME. Encouraged by the superior performance of combination treatment in vitro, we then investigate its therapeutic efficacy in post-ablation tumor models. The combination treatment significantly slowed the growth of residual tumors, with one mice survived more than 60 days and 50% of mice being cured. Then we wonder whether combination treatment would have certain effect on the aggravated immunosuppressive phenotype of residual tumors. Immunofluorescence results showed a significant abundant expression of CAR-T and HMGB1 after combination treatment, indicating a marked immunogenic death was produced. Meanwhile, the combination treatment blocked the expression of P110 gamma protein, thereby inhibiting the target that regulates myeloid cells infiltration. Further, flow cytometry displayed that the combination therapy established an inflammatory tumor immune lead, thereby exerting potent anti-tumor immune activity. Taken together, relying on a well-established post-ablation model, we offer direct evidence that incomplete microwave ablation induced tumor outgrowth related to a local complex inflammatory reaction characterized by hypoxia and mobilization of myeloid cells. Then, we rationally designed a nanomedicine with efficient tumor homing capacity, TME-responsive drug releasability, and in situ oxygen production properties that can enhance local radiotherapy. Specifically, this radioimmunotherapy strategy could effectively reshape the inadequate ablation-induced immunosuppressive TME and suppress locally residual tumors recurrence post-ablation. And that's the end of my presentation. Thank you for your attention.